Good morning, everybody. Spring is over in Scotland and we've gone straight back to winter. It is absolutely freezing. I've got a really good feeling about this week. My molecular cloning has been confusing a few times in a row now. I'm really struggling to verify if my insert got in, but I went back to the previous step and I double-checked the sequencing data and it definitely looks correct. So it's not that we missed a previous step. There's just something going wrong here to work out. So that's this week's job, and I reckon I'll have it sorted by the end of the week. Other than that, my fax data on last Thursday was really quite low, but I also didn't have enough cells. Looks like maybe I suctioned up some of the cells in some of the samples, but luckily I had another plate of those to take off for sequencing. Instead of sending them for sequencing, I'm gonna grow them out a bit more, take them for fax again. So I'm gonna get a second lot of fax results from that same lot of transfections. I've planned it all out in my lab book. Looks really good written down on paper. Let's see how it goes in real life. Good afternoon, everybody. So today's results were disappointing on one hand. Basically, my gel results just keep indicating to me that something went wrong in the last round of cloning. So I'm gonna have to go back and repeat that step, but at least I finally decided that I am gonna go back and repeat it. No more doing the same thing and hoping for a different result. So tomorrow will look different to today. So I'm looking forward to having another crack at the molecular cloning. I don't know what went wrong last time, so I'm just gonna do it again. But watch everything very carefully, leave my enzymes in a bit longer, that sort of thing. This afternoon's job is to explain the whole molecular cloning process a little bit better in my 10-week report. I kind of glossed over it, but seeing as it's half of what I'm working on, it should probably be included in the report. Alrighty, I'll see you guys tomorrow. Good morning, everybody. It is Tuesday and I'm heading in this morning for the first time. I have a feeling today is going to be a 2-entry day. So I'm going in early this morning to split my cells. I want to do this as early as possible because I actually want to use some of these cells tomorrow. So I'm trying to give them as much time as possible to grow. But I couldn't actually see any space at the benches this morning. So that's where I would do my molecular cloning. So instead of getting them both done at the same time, I'm going to go in and do my tissue culture work this morning, go home, watch a seminar, come back in, do my molecular cloning, and head home. The other good thing about this plan is that I'd like to send one more draft off of my 10-week report to my supervisor, and it's not quite ready yet. I was up late last night trying to get the figures together, but there's still a little more work I need to pull it all together. So that means I get time this morning, so hopefully I get it off to her by lunchtime instead of by midnight. All right, we've got a jam-packed day. Let's see how it goes. Good afternoon, everybody. Today has been a fantastic success. We're back on track with the molecular cloning. I got my tissue culture done this morning. I decided they didn't need splitting, so I just refreshed their media and they'll be good to go for tomorrow and the day after. My other job today was to get back on top of the molecular cloning work. So I'm going back to the previous 2 plasmids that I needed to cut up and stick together, essentially. So I thought maybe the problem was that one of those original plasmids, there was something wrong with them, but I just digested them and ran them out on a gel and they look good. So we're starting with good foundations and we'll keep building for the rest of the week. By Friday, I should have them on a plate. This afternoon, we have a seminar that my supervisor runs. We have one every 2 weeks, and I find it's a really good way to stay on top of what everyone else is doing in this area that's not in my specific niche. So I'm looking forward to my afternoon activities. I'll see you guys tomorrow. Good morning, everybody. It is a windy Wednesday morning, and I am heading in early. I'm hoping to get my digestion out of the way this morning, because then I will be preparing for FACS with my second FACS analysis of the old transfections. And then I also need to do a new set of transfections. The 5th time I'll be doing CRISPR in this PhD. This afternoon, one of my supervisors is giving a talk partially to do with my project, so I'm really excited to see that. So I want to make sure I get as much of this out of the way as possible so that I'm definitely free to listen to his talk. See you in there. Never mind, we're heading home. I hung up my key card. Alrighty, we are back on track. Yes, I have completely negated any time that I gained by waking up early. But my watch is quite happy with me. I'm at 4,000 steps and counting. So from an exercise point of view, I guess I'm doing really well this morning. I just didn't plan to be measuring this day by exercise. Alright, in we go. Attempt 2. Good morning, everybody. It is Thursday, and I am on my way into the lab. Today is basically a repeat of yesterday in that in the morning I'm going to be doing FACS, but I don't have all those other things to do. I do not have a transfection today. I don't have an onerous amount of molecular cloning work to do, so today's a lot more simple. Alrighty, see you later. Good afternoon, everybody. I got everything I wanted to do today ticked off, including working out what was going wrong with my FACS results. Actually, that's not true. I haven't worked out why it went wrong in the first place, but I seem to have been able to reset my settings, and now I can look at my data again, which is great. I don't think I really explained that at any point, but last time I went down for FACS, I wasn't able to export any data. All of the nice gates that I'd set up had been removed, kind of, but not really. They did still seem to be there, but I just couldn't see them. So it's annoying to not know what caused that, like if I clicked the wrong thing at some point, or if it was just a bug. Either way, I think I roughly got it sorted today, at least enough to see my data. This is certainly like a frustration I have in science, that I'm not a FACS expert, but I need these results to do my work. But it's not just FACS, it's also microscopy and fluorescence imaging and sequencing and coding and all sorts of things. There's a lot of elements to what I do, and to properly understand the results, I need to be an expert in all of them. But I can't develop all of that expertise in one go. Someone's filming something over there. Their setup seems a lot more professional than mine. But anyway, I'll see you guys tomorrow. Good morning, everybody. It is a grey and wet and windy Friday. I don't have much to do in the lab this morning. Yesterday, I set up some plates for overnight cultures that was made up of transformed cells that had hopefully the little plasmids that I made in them. So I need to get those out of the incubator and into the fridge so that they don't keep growing. I want to keep them at a single colony stage where they're not starting to grow out and merge into other colonies. Because next week when I come in, I want to be able to grab just one colony. So yesterday I set up some plates with the transformed cells that had my ligated plasmid inside of them, hopefully. And this morning I need to get those plates out of the incubator so that they don't overgrow. I also need to do some basic tissue culture. I need to split down one plate, refresh the media in another one probably. But other than that, I'm going to spend the rest of today preparing for lab meeting. So over the last 2 weeks, I've got a lot of data, so it's definitely time I present it at lab meeting. I've got some of it typed up, but not that FACS data from tomorrow, which is really the bulk of my data. So hopefully I get all my lab work done in time to get home, get that sorted, work out where in my house has good internet, because I've been really struggling with internet recently, and then present at our lab meeting. See you guys later.
So next we're gonna head downstairs and I'm gonna check out the transformed colonies from my ligations, which I did like yesterday and the day before. On plate 1, I'm expecting just a few colonies. Plate 2, I'm expecting more colonies. On plate 3, I'm expecting no colonies at all because theoretically the plasmid should not have been able to ligate back together. Therefore, my colonies can't be expressing the ampicillin resistance. Therefore, they should not be growing on this agar media with the ampicillin in it. So let's take a look. On plate 1, we have just a few colonies. You can kind of see them in the light. Excellent, looking good. Plate 2, we have more colonies. Excellent, just as I expected. Plate 3, we have more colonies again, for some reason. Great. I'm probably gonna have to do this again. So this is now 4 days work. I'll be repeating it next week.
Hello again, everybody. I made the mistake of thinking that the rain had stopped and therefore it would be warm enough to not wear my jacket, and I was wrong. I'm freezing, but if I stop to put it on, I'm just gonna get more cold. I'm nearly home, and also then it started raining again. So anyway, there were some really weird results in the lab there. It's really frustrating. I wish I'd filmed yesterday now, because then I'd know for sure exactly which tube I used. I remember looking at them and checking them and doing it. It's not like I was on autopilot. I do remember doing it, so I don't think that's a mistake that's happened. So I had a chat with my supervisor and we're pretty agreed that I just need to do it again. So my lab work is done for the week and I've got a very clear picture of what I'm doing next week. Now I need to go home and make sure I've got a good, tidy presentation of my data for a lab meeting in a few hours. See you guys next week.